Test, test. Can you hear my voice? Okay, thank you. Good afternoon, everyone. Let's wait for a few minutes for others to join the call. Today, we will continue to learn about functional genomic computation tools. My focus is to introduce you as many tools and as many pipeline that you can reuse for your research. And especially for functional genomic, there is many tools, frameworks developed in R language or Python language, but we will focus on Python this time, and few tools that is selected this semester. Yeah, if there is any other better tools, we will introduce also, but we will focus on TerraBio. TerraBio you already experienced before. I hope you can reproduce your first pipeline in TerraBio using GATK. TerraBio is a well-known tools for Illumina devices. TerraBio is funded by MIT Broad Institute and Illumina and some technology principal like Microsoft, Amazon, and Google. Okay, let's start this week I want to introduce another library, and then I will hand over to Sinta and Fadin to introduce low-code computation platform called Galaxy. Okay, so previously we learned about Biopython. So Biopython is a very famous library, collection of APIs that you can use for many topics. Please download the documentation and try your prompt engineering. Biopython is commonly used tools in Python. You can download this book in PDF. You can print if you like, or if you like to read the PDF, you can just read the PDF. So many operation in the sequence object. Sequence is ATCC, all of this sequence of the proteins inside DNA and RNA. And you can do many tasks inside the sequence, like slicing, turning sequence into string, combining or concatenating, find out nucleotide sequence and its reverse complement, transcription, translation, making a translation table, compare the sequence object, and many more. You can also do annotation like doing the sequence record object, annotate the sequence record from stretch or from FASTA file or from NCBI or ABI gene bank files. You can do features location and object positioning. You can do sequence input and output, alignment, and many more. So this is very important library that being used by many other library, higher level library, because there is many API and functionality from exploratory analysis to unsupervised learning is implemented in here. Okay, so spend enough time for this important library, and also tools that will be introduced by Sinta and Fadin. And today I will introduce another library, yeah, called DE-Sequence2. So this library initially implemented as a R library using R programming language, but community have ported the tools, this library, this package, as a Python library from original R. So they call it PySequence2, PyDE-Seq2, yeah? So if you open your Colab, you can install these tools. using pip install, you can just install in Colab or Kaggle. Pip install PyDE sequence 2. Sorry, I'm worried if I'm wrong. So PyDE sequence 2, yeah. Okay, and then you install. Wait, it's still connected. So what is this? Biopython usually working with DNA sequence, and this one is mostly with RNA, yeah. So it's a package ported from R, yeah. So it's not a big package like Biopython, but it's very useful for RNA manipulation. So there is a 3 tutorial here, but the best way to learn this is to go to the GitHub, yeah. If you go to GitHub, you will see the source code, yeah. First, you need to pip install this until you successfully install. And then you can go to this folder in GitHub to see some data that you can try to manipulate, yeah. There is test count data. So you can download. The file name is testcounts.csv, and then you can upload to your which is okay. You can upload to your Google Colab. So once you upload, you will see the data in testcount.csv, and then you can try some tutorial in the code, yeah. So here, you can go to the documentation, there is getting start, and you can see the first tutorial. Okay, so let me copy paste here. So the first code is like import the library, yeah. You need to pip install before it works. And then you have to make your output path. It will be written to this Google Colab, the output path, yeah. And then let's see the tutorial, what the tutorial is trying to do, yeah. So DE means differential expression analysis, yeah. So it requires 2 types of input, the count matrix, the number of sample and number of gene in that sample, and read counts, and also some metadata. So here it will give you the metadata as well. You can download in case you need, and then you can upload the metadata as well, okay? And then let's read the data, okay? I think we don't need this code, just asking ChatGPT, I have a file, okay? Because the file is already there, it test_counts.csv. I have the test_counts.csv in Google Colab folder. Write Python codes to read the data and load into, and you have to see what kind of library it used to load the data. Here it used pandas, PD, yeah? So PD read, so using pandas. So you have to say that I need the code to read that file and print the file, print the data frame, yeah? So you can say pandas data frame, okay? Okay, you can just pass the code, make sure it used the same code, yeah? So from Google Colab, import content drive, and then test count, okay? And then you see the code before it said counts underscore df. So you say counts underscore df. And then here also you have to change this counts underscore df. Connected. It's not Google Drive, yeah? It's not a Google Drive. So wait, I'm wrong here. Okay, just directly read the file. I just have to remove this one and then try to read again. Yeah. So you get the sample data and then you can continue this. Okay. You have to do yourself to make sure that you're familiar. And I will explain what is this. So this is the gene and sample. How many counts it have this gene in the sample. Okay. So you also have to download the metadata and then look on the metadata sample and the code. Usually metadata is containing some condition and grouping, yeah, for the in the annotation. And then you can start pre-process the data, like display the metadata condition, count the data, okay, and this one, and you can do analysis like single factor analysis in this example. So this is the code. It creates the differential expression sequence dataset, okay, DDS, and it print the DDS, okay, and then you can save the file using Pickle. Pickle is another Python library. And then it performed the DDS farm, like showing the access dispersion and LCF. And you can complete this tutorial. There is 3 tutorials. There is another tutorial is using the same data set, and you can perform step-by-step workflow. Okay, this workflow is first read the counts modeling, perform the computation normalization factor, gene-wise dispersion, dispersion trends coefficient, etc., and perform some statistical analysis of the data like Wald test, Cook's filtering, p-value, and LCF, LFC sync rates. Yeah. Of course, you need to understand statistically what is this test, like Wald test, Cook's filterings, and p-value. Usually, this is hypothesis test in statistical term. Okay, so try this. Usually this Python library is utilized by another genomic Python library like SnakeMake. We will learn about SnakeMake later, but SnakeMake can be combined using Biopython and also differential expression library. We will learn later on about SnakeMake after we learn about Galaxy, yeah. So today we will continue learn about Galaxy, but before you continue, I believe it's good for you to learn about Biopython and also differential expression library like PyDESEC2 library. Okay, that's it from me. So please continue with Fadin and Sinta to learn about Galaxy. They will introduce you what is Galaxy project. So it's an open source community project, drag and drop for workflow. You can reuse many library predefined workflow from Galaxy, and you don't have to install anything. You just use Galaxy. Go to usegalaxy.org. This is installed in the cloud. So what is this? It's a tools. Yeah, drag and drop tools to get data, send data, do some text manipulation on the data like pre-processing, working with genomic file manipulation like FASTA or FASTQ, SAMBAM, VCF, BCF, including Nano4. because GATK is too much focused on the Illumina. So this one is more open for Nano4 and other format. Nano4 is another gene sequencing devices that is widely used currently, other than the Illumina gene sequencing, and a lot of things. And you can find papers in bioarchive that utilize Galaxy. For example, this is a biology archive paper. Okay. So like this one is a transformer-based tools recommendation in Galaxy. Yeah. Plant Describe is a tool for comparative gene and family analysis in plant genomics. Okay. There's a lot of paper here still utilizing Galaxy. It's good because if you try to find topics with Galaxy for your master degree, you can find the latest paper and latest use case, how people use this Galaxy. Okay, so this is, for example, from bioinformatic groups in Germany. They build transformer-based tools recommendation system in Galaxy. You can download, you can try. Once you understand neural networks, in the previous lecture we talked about neural networks, you can use feed-forward neural network and embedding for making recommendation engine for genomic. And I think what they implemented here is they implemented it using Galaxy. Another paper here that I want to show you is plant types 2 is plant genomic. So it's not human genomic, I guess. So it's a plant genomic for family tree of the gene, plant gene. Okay. Single cell RNA using Galaxy framework. Many more training infrastructure services. No, not the one. Multiomics. This one is also plugins for Galaxy. Okay. So Galaxy is quite widely used in bioinformatics community. So I think it's worth for you to spend time, maybe 1 or 2 weeks with Galaxy before we move to code-based tools like SnakeMake and Nextflow. That's it for me. Please continue. Is there any question before continue with the Calbe team? Okay. Okay, if no question, please, Sinta or Fadin, please start introduce Galaxy.
All right, thank you, Parisman. Kashinta here seems to be having some technical difficulties, so I'll be stepping in for her. So I'll be sharing this my screen. And here we'll be using 2 links. I'll just quickly send them over by chat. So the first link is the Galaxy platform itself, and the second link is the tutorial we'll be using today. So here on my screen, we have 2 windows. The left one is the Galaxy platform, and the right one is the introduction. So the first things that we're going to do is we'll be having a quick overview of what we'll be discussing in today's practical session. So first of all, we're going to start with how to get started in Galaxy. We'll be learning things like how to upload a file, how to use a tool, how to view results, view histories, extract and run a workflow, and how to share a history. And just like Parisman said earlier, Galaxy is an open source web-based platform for bioinformatics analysis. So Galaxy enables users to access a wide range of tools, which we'll be exploring later on. And since this platform is very user-friendly, we don't need to code anything in this platform. Okay, so going back to the tutorial, is everyone already opening these windows on their own screen? Maybe If you guys are having any difficulties, you can just open your mics or send a message in the chat. All right, so the first thing we're going to do is we're going to log in. Here in my screen, I'm already logged in, but for demo, I'll just sign out again. And then we'll be met with a login page. For those of you who have previously made Galaxy accounts, you can just log in directly. But for those of you who are new here, we can just create a new account. So you need to input your email address, your password, and a public name that will be shown in the Galaxy platform. So once that's done, you just create your account, and then we can just log back in. And once we're in, you guys will see this page. And here we have 3 panels. The left panel is it's called the tool panel. And then the middle panel here is the viewing panel. And the panel on the right is called the history panel. For those of you who are new in using Galaxy, your panel will probably look something like this. Unnamed history. I had a previous history because I've been working with other histories beforehand. So I just created a new history. And then the first thing we're going to do is we're going to rename that history to my analysis. All right, so once we've renamed the history, we click the edit button here. And then we type in the new name, my analysis, and then we just click save, and then our history is renamed. All right, so is everyone following along? Is there any difficulties? I guess we're all good. All right. So the first thing we're going to do is, when we're doing an analysis, is we need to have data, obviously. So here we can upload the data by going over the Tools panel on the left. And there's this Upload Data button, which we can just click. And in Galaxy, we have several options in uploading data. We can choose a local file. We can choose remote files, or we can paste or fetch data. And in this tutorial, we'll be using the paste and fetch or fetch data. So we'll just go over and click this. And in this box here, we'll be copying the link provided in the tutorial. We'll just go ahead and copy that. And then we'll paste that link in this box. So once that's pasted here, we can go over the Start button and click that. And we'll just close this window. And here the job is already executing, it's still running. We'll just wait for that to turn green. So once it's green, it's already completed. So the idea here is that this is a bacteria FASTQ file. So it's a sequence with quality scores. And here, since the job is already green, we can just click the display icon here, the one that looks like an eye. And this might take a while. There. So as I said, this is a FASTQ file. And here we can see that the first line here is the sequence identifier, and then the second line here is the sequence itself. Sorry, and then here we have the quality separator or the plus character, and then on the fourth line is the quality scores. So the quality scores, just so we're on the same page, they're an indicator for the confidence or accuracy of the base calls in the sequence. And this will be used as a basis for analysis in our next tool. So if we were to just take a look at this file as it is, it's a bit hard to get any insights just with this raw data. So the next tool is we'll be using a tool called FastQC. To access this tool, we'll go back to the Tools panel, and then there's a search bar up here. We'll just type FastQC. All right. Once we've typed FastQC, there's this tool on top, and we'll just click that. Okay. So in this tool, panel, the viewing panel, there are a few parameters we can control, but in this case, we'll just be using the first one, which is the raw read data from your current history. And here in the drop-down, it shows the data we can use. And since in this tutorial, we only have one data here, which is the mutant R1 FASTQ, the default will be that exact data. And all we have to do now is just to execute this tool, we just click this button up here, Run Tool. We just click that. And we'll just wait for the job to execute. So this tool produces two outputs, raw data, which is text-based, and a web page, which is the visualization of the raw data. So basically, this is a summary of the FastQC report, and it's used to assess the overall quality of the sequencing data. So for the raw data, I can show you by clicking this I button, this display button. And here we have a very long text-based data. And for the visualization will be continued by Rashidan. So go ahead, Rashidan, take it away.
Okay, so the next one is we showing the visualization of results. So as you can see, right. So as Jacqueline mentioned, there is raw data and web page. Raw data is like this one. So it's raw data, and there's like a mean, median, lower, and etc. And there is a web page that contains a visualization of data. So you can press this button that show display. So there is like a 6 statistic, and there is a per base sequence quality like this, and there's like a quality score plot per sequence like this, and there is per base sequence content, per sequence QC content, and per base and content. So the summary is of if you want to see the visualization or plot of the data you imported, you can press this web page one, and if you want to see the like table one or maybe just the numeric of it, maybe you can click the raw data one like this. Yeah. So the next one is run other tools. So we will, after we using a FastQC, we will use another tool called filter by quality. So you can type this in here, filter by quality. So this tool is make sure that our FastKey file is filtered. So the lower quality file will be filtered, and we only contain high quality data in our report. So there is a parameter called quality cutoff value and present basis in sequence. So the quality cutoff value is a threshold value that determines which rates are considered high enough quality to be kept and which are low quality and should be filtered out. In this case, any rate with quality score less than 35 will be filtered out. And there is a percent of base in sequence that must have quality equal or higher than cutoff value. That means that must have quality equal. That means that it's a bit more complex actually. So this parameter is used to determine the percentage of bases in array that must have a quality score equal to or higher than quality cutoff value. In this case, this parameter, which is 80% of the base in red, must have a quality score equal or maybe higher than 35 in order for the rate to be considered high enough quality to be kept. So let's just change the parameter as according to tutorial, which is 35 and 80%. 35 and 80%. After this, we click the run tools. Okay, this one. So it's actually same. So then we wait for the history for changing the color into green. So we will wait. Okay, so the history already turned green. So we could see the output of data. So here's the report result of fast key sheet that filtered out, which is the quality score below 35 will be filtered. And there is like best percentage of 80% from the 35 score. So that's it. So the next one is, so we called changing the parameter too in here from history, but we should make sure that our parameter is right, which is 35 and 80%. So we could click this button, the green button. So we could see that quality cutoff is right, which is 35 and minimum percentage is 80%. To change the parameter, or maybe we want to reload another run with a different parameter, we could press this reload button and we could change the parameter here. So in this tutorial example, we will use 36. So we will change this to 36 and we run the tools again. So there is a new history on here. So this is like this one used 35 score and this one used 36 score. So it's already turned green. We could see again. So this one is using the quality cutoff of 36. Yes. Okay, so the next one is share your history. So we will try to share our history to another people maybe. Yes, so like you can click history options, the gear icon, and tap the share or publish. So like here. And this one. Yeah, you can input someone like your friends or maybe collaborator that want to join the workflow, and you can specify to email. For example, if you want to send to this one, you can add like this. Oh, so like maybe another person. For example, I just put a random email. So yeah, there is, so you can share this workflow with another person. And the next one is convert your analysis history into workflow. So you could also turn your this history one into a one wall or workflow. So you can just use extract workflow from history. So like this one. So this is the workflow we will want to turn into workflow. Okay, so we will change the workflow name to something more descriptive, such as key and filtering. So we will change the name. And we want to rename the workflow input in the docs in the box at the top of second column, which is to FASTQ reads. Maybe like this one, like this. And after that, we could create the workflow from here. So yeah, the workflow is extracted from our history. And now we will create new history, then we convert our workflow into the new history. So from this history option, we create new history. Okay, from here, the plus symbol in here. After that, we want to copy our workflow into this new history. So first we will change the name first to next analysis. Yes. After that, you will want to show history side by side first to copy our history from the previous one. So press the show history side by side. And here is the new history, which is called next analysis, and then previous analysis, which is called main analysis. So to copy our history, we could just drag from the previous analysis into the new one. So let's try it out. For example, this one, the input data, we just drag this into here. So yeah, it's done. Our history copied, which is the mutant data. And now we will into the last one, which is run workflow in the new history. So first we press the workflow in the top menu bar of Galaxy. So this one, click in here. So yeah, there is like, we call the previous workflow, which is called key and filtering. We will press this because previously I already tried to make some new workflow. So there is like two, which is the same name. So yeah. So we press this one, which is the recently made workflow. Click this. Click the run workflow, and we will wait. So it will convert it as history first, yes, like this, and we will wait until all of them are turned into green. We will wait. Okay, so all of the history are already turned into green. So the workflow is already imported here. So we could view the report and generate PDF too. So we will try to see it. There's also detail, which is our input data called mutant or ASCII rates. And there's also steps, which is we imported from previous analysis, like FASTQ reads or maybe input data. And step 2 is like generating FASTQ reports. And step 3 is filtering by quality, which is 35 score and 36 score. So we could also turn the reports into another object, like a biocompute object, which is an official name for JSON object for bioinformatics, maybe also another metadata too. So let's see how the PDF look like. So here is the output of workflow, which is FASTQ data that are filtered. So like this. So it is so long and sorry. And there is also a workflow step 2 in here, which is FASTQ read until the last steps. So this is the view report. If you want to view report, not using PDF, you can see in here in the website. So just click the view report one. So there is like, this is the output of input data, which is are returned into report and filtered. Lower quality, the lower qualities are defiltered. And this is the workflow output, such as our lower score filtering and best percentage and etc. So that's it from me, and the tutorial is already done, and maybe we could change into conclusion, which is will be provided by Jacqueline. Can you show the conclusion maybe?
All right. So thank you, Rashidan, for your section. And we'll go back to the conclusion. So what we've just learned today, we've learned how to open the Galaxy platform. We've learned the panels available in Galaxy, and then the actions we can do. We can rename a history, we can upload a file. We also use the tool. We use the FastQC tool, and we were able to view the results from running that tool. And we also filtered the data based on their quality scores. So we've also used another tool, and we learned how to rerun a tool. So with the same tool, we just rerun that exact same tool with different parameters. We learned how to share our history and also create a new history. And we also ran the exact same workflow in that new history. All right. So I guess that is it for today's practical session. Maybe if there are any questions or any concerns, you guys can voice them out by unmuting or in the chat. You guys can type out your questions in the chat.
Thank you, Jacqueline. Thank you, Rasidan. Yeah. So I think it's a good introduction as an eye-opening for the Galaxy tools. This is similar but more advanced than TerraBio because you can drag and drop setting parameters. But of course, there's a lot of workflow, yeah, a lot of features. So the best way for you to learn about these tools is to reproduce the working workflow. So please do some literature review to find out as many as you can papers that reproducible. So it's just like AI engineering discipline. We rarely develop the workflow from scratch because we improve from the previous researcher who are working on this for many years. So make sure that you find workflow that is credible, reproducible, because in bioinformatics community right now, most of the people focus on FAIR principle. First, the workflow have to be findable, like other people can find it. And the second one is the workflow should be the A is accessible, means that you are not closing your workflow only for you, because this is about human or about biology, right? Sometimes about critical illness. And next is should be reusable, means that other people don't have to redo what you have done. So other people can reproduce what we have done. And the last one is we can interpret correctly the workflow. Okay, so FAIR principle usually being adopted by whole bioinformatician around the world, and your job in as a student in University of Indonesia is to find out papers that use Galaxy and then try to reproduce the workflow that they are developing using Galaxy workflow. Okay, the same discipline you will have to do for SnakeMake and also for Nextflow to make sure you have catalogs of reusable workflow for the tasks that you are doing in functional genomics. So that's it for today. If any question, please don't hesitate to call to talk to Jacqueline and my team in Kalbe, Fadin, Sinta, and also Rashidan, and also on me in the group. We have a WhatsApp group for this course. And make sure you practice a lot because there's so many features in Galaxy that is very hard for you to memorize everything. So start from the working and validated workflows and reproduce it. But Galaxy have a very good tutorial. That is what Jacqueline showed and Rashida. You can start from the tutorial. There is also many tutorial in YouTube that you can do. Okay. That's it for today. Hopefully next week we can reproduce more workflow with Jacqueline and Rasidan, and the next following weeks we will do the same for other framework like SnakeMake and Nextflow. Thank you everyone. If no question, see you next week.
Okay, sir. Thank you so much.
Thank you.